All right, Professor Klein back here in the Human Anatomy and Physiology labs at Ohio University today to bring you lab discussion number 4 on the tissues, the skin, in the Semmes Weinstein Monofilament test. And if you've been following along the past couple of weeks, we've kind of worked our way from a molecule to a macromolecule to an organelle, a cell, and now we are on to tissues and how tissues make up the body, form organs, organ systems, and essentially the entire body itself. And as I go through the PowerPoint today, definitely be looking at your lab manual, filling in a lot of the stuff within that manual, and we'll be talking a ton about where these different tissues are actually at inside the body. But before we do that, we got to talk about how you use a microscope and some of the things to be looking at when we talk about histology. Now, histology means the study of microscopic structures within tissues. So that's like taking one of these slides, and this one's got the thymus on the slide. So they cut part of the thymus out of the body, or you could cut part of the heart, skin, whatever, and you'll be able to see it on this slide. All right, and histology means the study of microscopic structures within tissue. So for example, on your screen, let me make my box smaller here. Now you can see something like the lung tissue. You take a small slice or chunk of the lung tissue and you put it onto this slide, super small, all the small things, right? Super small. Take it, bring it on over, put it underneath the microscope, and then you can blow it up and see a picture like this right here of lung tissue. But I'll tell you, what you see underneath the microscope is not always the same color. What they do is they dye a lot of these tissues purple, pink, blue, red. You know, they take a little bit of dye like this and they put it on there so that it highlights certain structures and are easier to see. And if you're looking at one slide, let's say I'm looking at this one slide here, you might have multiple different types of tissue within one histology slide. For example, this part up here is epithelial tissue. This part in the middle, let's divide this here. This would be like smooth muscle. And then down here more maybe some connective tissue. So you're going to see different types of tissue on one singular slide. And lastly, not all slides look the same. They will look similar, but you want to look at a variety of slides to get a full perspective of that tissue. And what are the tissues of the body? Well, let's look at the 4 main categories of body tissue. We're talking epithelial tissue, which, let me close my box up here and you can see that's like skin. That's like the lining of the body, as well as the internal lining. As you can see here, inside the intestines would be some epithelial tissue. So keeping things out and keeping things in the body is epithelial tissue. Then we've got connective tissue right here. Tons of different type of connective tissue. We're going to spend the most time on this category, but as you might have guessed, it connects different structures in the body for support and structure. That's connective tissue. Then the third category is muscle tissue. We've got skeletal, we've got cardiac, and we've got smooth. And we'll look at where those are at in the body as well. And lastly, nervous tissue here. This is a neuron, make up nerves in the brain of the body. We'll definitely look a little bit at nervous tissue. Let's get things started with epithelial tissue. And the first thing you got to ask yourself with epithelial tissue is how many layers is it made up of? And we're going to name it after how many layers it's made up of. If it's got one layer, then we call it simple. So one cell thick is simple. 2 or more, we're going to use the word stratified. So if you were to draw these out, and I would highly suggest drawing these out, you got spaces in your lab manual to do that, so definitely draw these out. but this is one cell thick up here. That's simple. Down here, we're looking at 1, 2, 3, 4, 5, 6 layers. That would be stratified. But the second question you want to ask yourself is, what are the shape of these cells? What are the shape of these cells? If it's squamose, then we're talking about like a squiggly egg. It might be flat, it might be rounded like that. These are all examples of a squamose type cell. Now cuboidal, you guessed it, just a cube. So square, cube, that's cuboidal. Columnar is a tall column, like a building type structure would be columnar. And then lastly, pseudostratified. Let's take a look at that. That's a special one in the shape. All right, here I am on APR, so pull that up if you don't have that up yet. And essentially we're looking at pseudostratified columnar epithelium. So it's got the word columnar, so it is like these tall column-shaped cells. But as you can tell, sometimes there's cells in the bottom, sometimes they're kind of smashed together, and most often you'll have these cilia or hair-like projections coming off of the top. So cilia increase the surface area of this type of tissue, and then the pseudostratified columnar epithelium gives it a distinct look and shape. Let's continue our comparison of simple versus stratified. So here's a look at squamous, cuboidal, and columnar as you go down, but on the left you've got the simple, on the right you've got the stratified. Now notice all these thick layers on the right, all these thin layers on the left. Now a key phrase that you'll want to know for anatomy and physiology and progressing forward as a medical professional is the phrase function follows form. When you say function follows form, that means whatever the form is, whatever the anatomy is, the function typically follows that. The function is dependent upon the anatomy. So in this case, it's completely true because the function of the thin, simple tissue is to exchange nutrients and waste and gases, just like the diffusion we've talked about in the past. And that would make sense because one cell thick is like a one piece of paper. So grab a piece of paper wherever you're at and take a pencil and try to poke through that piece of paper. I'll give you a second. Grab a piece of paper, try to take a pencil and poke through it just like a cell or oxygen or nutrients would go through this simple squamous tissue. So, all right, you ready? Here we go. 3, 2, 1. Boom. Easy to do. Pencil went through. It's thin. But now grab a book. Maybe your lab manual right here. This is super, super thick. This would be like your stratified squamous. There's no way, as hard as I try to push this pencil through this lab manual, there's no way I'm doing it. So stratify is more about protection and secreting things. Not a lot of exchanging or diffusion going on with stratified. That's more the simple. So remember that as we continue on here. Here's another view of the pseudostratified epithelium with the cilia on top. But when you're talking about epithelium, you've really got to break down the superficial and deep sections of this. So what this would look like in the body is, let's say you're in the intestines, right? We got a Dairy Queen Blizzard, we got a hot dog in there. Let's go in through the intestines that I said earlier is where you might find this epithelium. Now we can say, okay, the top part of that epithelial layer is called the apical surface. So apical is the free surface. So that would be like this layer down here and then this layer up here. It's facing the lumen, or it's facing the inside of the intestines. Now, if it was your skin, it'd be facing the external environment, but it's the free surface. Now, furthermore, deeper, we've got something that's called the basal surface. Basal surface would be right here. It's right next to the basement membrane, but this is a deeper layer, and it's talking about the fused layer, the layer that's attached to the deeper structures with that basement membrane, as you can see right here, smashed up against that side of the epithelial tissue. And even underneath that, deeper to that, you've got connective tissue, which you can see down in here. So we want those to be tight and junctioned together, and that is how they do that. And if we look at some real life examples here, this is the gastroesophageal junction. You will see this again in BIOS 1310 when you talk about the digestive system. So get this one down now. This is the junction between the esophagus and the stomach. And when the esophagus comes down, it's got stratified squamose, because you don't want to absorb things in the esophagus. You just want to transport them down to the stomach. Now, in the stomach, you got a little bit of absorption going on. So it switches right about here, and that's the gastroesophageal junction, to simple columnar. So notice how you go from this thickness to up here, maybe 1/4 of the distance. So 1/4 the length for the epithelial tissue, because now it's a simple columnar. You can also see this on skin when we talk about the different layers of, let's say, the epidermis here. You can see one top layer, which is really thin, and then you've got a thicker layer down in here, and then it finishes with a thin layer. We're going to look at this much, much closer, but that's just a few examples of epithelial tissue. So pause the video if you need to right now and reveal epithelial tissue, because we're about to get into the skin and apply a lot of this information. So pause the video if you need a second, take a short break, and then come back for some more. All right, let's jump into it here with the skin. And with the skin, there is a YouTube video summarizing everything in this skin model. There's a lot packed within this model, so check that video out before you go any further in the PowerPoint slideshow. All right, welcome back from that skin model video here. Let's break down a few of those things for you. First off, the epidermis. That's largely what we're going to focus on first. These top 5 layers here would be the epidermis. If it's 5, it's thick. If it's 4, it's thin. And as we get down into the dermis, you can see a lot of different structures within there. And then finally, the hypodermis down in this region. So hopefully you got to watch that video as we move forward. And let's start by generally talking about what skin is and how it differs throughout the body. Like I just mentioned, there's thick skin and thin skin, the 2 different types of skin. Now, thick is going to have those 5 layers. It's got 1 extra layer because thin only has 4. And if you're thinking about where this is at on the body, well, we've got the palms of the hands right here, and then the soles of the feet. I'll just show you a picture of those right here on the left. All the functions of skin are over here. We're going to do protection, containment, heat regulation, sensation with a lot of these receptors in here, as well as synthesis and storage of vitamin D. Speaking of the sensation of the skin, I've highlighted 2 of the corpuscles, the Meissner's corpuscle, which is this right up in here, and the Pacinian corpuscle. Now, if you were to describe this, you'd say, okay, the Meissner's corpuscle is deep to the 5th layer of the epidermis, or you would say it's superficial to these other structures here. Regardless of how you describe it, my question is, what does the Meissner's corpuscle detect? Second question is, what does the Pacinian corpuscle detect? What sensation? Take 10 seconds, find the answer, think about it. What does the Meissner's corpuscle detect versus the Pacinian corpuscle? All right, if you said the Meissner's corpuscle detected light touch, you would be correct. If you said the Pacinian corpuscle detected deep pressure, you would be correct as well. But here's how that happens. As you push in on the skin, you have to deform a corpuscle or receptor. Deform means to change its shape. So if you only push in a little bit, like let's say a mosquito lands on your arm and it pushes in your skin, it probably will only reach this layer of the skin, which has the Meissner's corpuscle, which tells you you're feeling light touch. But let's say somebody really pushes down on your skin, it'll probably reach and deform the Pacinian corpuscle, and thus you'll feel deep pressure. So a little bit of physiology with the anatomy here of the skin. But let's zoom into the 5 layers of the epidermis for thick skin. So we're looking at thick skin here, and really you can see first off the stratum corneum. So stratum corneum would be anything from that line I just drew upwards. Very, very thick, lots of time dead cells. If you rub your hands together here, you'll get that stratum corneum falling off of your body. Now next up is the stratum lucidum or lucidum, and that is like the layer between the black lines there. Stratum lucidum, it's a clear layer. It's only found in thick skin. This is that extra layer. It's only found in thick skin. So lips, palms, soles of the feet, that would be the stratum lucidum. Next up is the stratum granulosum, and this one's back to a thin layer. This is just this thin layer in here for the stratum granulosum of the skin. But then it goes thick again, some more stratified spinosum, and you can see all these layers down in here as the stratified spinosum. And lastly, the stratum basale is the bottom or deepest layer here. It is just these cells. So just these cells right here would be the stratum basale. And the word stratum doesn't mean stratified. So this is actually a single layer, simple, of the cuboidal cells. As you can see, they are cube-shaped. Cube is shaped down there in the stratum basale. And 2 specific types of cells that we want to focus on are melanocytes and stem cells. Melanocytes and stem cells. Melanocytes are going to produce melanin in the body, which deals with skin pigment. Let's look in the microscope and see what we can see of skin. So these are histology slides, lots of slides on APR for you, but here is thick skin. Now notice all those 5 layers. And here I drew some lines on so that you can see how that separates. Now notice the stratum lucidum is not as clear as before. That's all right. That layer is still present because this is thick skin. So take a second, see if you can figure out the 5 layers on the picture on the left. Try to replicate what I did on the right, but on the left-hand side picture. And this wouldn't be a Professor Klein lab discussion video without some study tips. So here's a study tip, or rather a mnemonic, to help you with the 5 layers of thick skin from superficial to deep. And that mnemonic is, come, let's get sunburnt. Probably something you never want to do is get sunburnt because that's actual damage to the skin. But it's a mnemonic that will help you remember stratum corneum with the C, stratum lucidum, L, granulosum, G, spinosum, S, and then basale for B. 2 more anatomical terms coming at you. The first one is the epidermal ridges. Epidermal ridges refer to the green part there. And really, these are ridges formed as you travel down from the epidermis. So over here, it would be like these parts for the epidermal ridges. I've highlighted it in green for you, but there's another very similar term called dermal papillae, and these are coming up from the dermis. So I've highlighted those in blue, and they are part of the dermis, but they are coming up to form papillae, or kind of like bumps within the skin. So if we were looking at the skin model over here, these would be your dermal papillae. Dermal papillae coming up from the dermis over here, but then coming down from the epidermis are the epidermal ridges. And fun fact for you, the epidermal ridges are actually responsible for your fingerprints. So these ridges, how they're designed and constructed, make up your fingerprints, and everybody has different fingerprints. All right, before I move on, I want to go clinically talking about melanocytes and skin cancer. So a very common type of cancer is skin cancer, dealing with the skin that we've been looking at here. But essentially what happens is overexposure to sun, or like a tanning bed, UV light can produce or stimulate the production of melanin, which is from those melanocytes. Quick knowledge check here. Which layer of thick skin are melanocytes found in? Which layer of thick skin are melanocytes found in? Or really thick or thin skin, this layer has melanocytes. Tell me in 5, 4. All right, if you said stratum basale, you would be correct. Stratum basale has the stem cells as well as the melanocytes. But melanin is not the cause of the skin cancer. You have the UV light increases the melanin, which is actually trying to protect your skin from damage. But in doing so, you have that overexposure, which can increase the melanoma cancer risk. But the risk is different in different skin colors. So let's take a global perspective at the different colors of skin around the world and relate it to the cancer risk. And as you can see from this chart taken from a research study that we'll look at, those with white skin have a higher risk of cancer versus the other types of skin color. So what would this actually look like on a person? Let's take a look. Now here are 3 examples of skin cancer. The 3 different main types. We've got squamous cell carcinoma, basal cell carcinoma, and just melanoma. Now you can see the deepness is what's different between those 3. However, if you're going to identify this, you want to do it early. As a medical professional, catching this early is very important. So over here is kind of an evolution of an irregular, asymmetrical skin cancer development in someone with white skin. However, if you wait too long, it could become very noticeable, and at that point, the risk of fatalities is very high. But let's do the global perspective and see how this looks on, let's say, someone who is Asian or Latino or Black. The identification, especially early in the diagnosis, is going to be tougher, especially with darker skinned individuals. And I'll close my box here so that you can see this research study that looked at cancer concerns in people of color, the risk factors, and prevention. And here's what they found. Essentially, it was found that those with naturally darker skin essentially have more protection against skin cancer or those UV lights. So they have less likelihood to be affected by skin cancer. However, they are much more likely, and I'll highlight this sentence here, to die from skin cancer because that delay in the early identification. So as a medical professional, you want to not only focus on the majority of people affected, which would be European Americans or white individuals in this case, but also especially the minorities that might have different symptoms or presentation of, in this case, skin cancer as compared to the majority. So check out more information from this article over here, or if you want to read this full research article, definitely check it out down here. All right, let's move on to thin skin here. When we're talking about thin skin, it's everywhere else except the lips, the palms, and the soles of your feet. So everywhere else would be thin skin. And we mentioned before, it's only 4 layers. So here are the 4 layers. I put 4 different colors to identify the 4 layers: corneum, granulosum, spinosum, basale. You'll notice there's no lucidium. So what I say is you lose the lucidum. You lose the lucidium. Either way you pronounce it, that's the layer that you're going to lose when you go from thick to thin skin. All right, that's the end of the epithelial tissue. So what I want you to do now is take a break, go through your lab manual, try to fill in those pages from epithelial tissue. Also, take a short break. Take 5 minutes, come back, get something to eat, come back, and then take a blank piece of paper and try to write out everything you remember about epithelial tissue. Take 5 minutes to do that. That's 10 minutes total. That will be extremely beneficial for your brain to learn this information. So do that now. Welcome back from that break, and hopefully you're feeling refreshed because we're on to connective tissue, and this is a large category of different tissues in the body. As you can see, there's connective tissue all over the body, but it's broken down into the 3 main categories. We've got the first one, connective tissue proper. Second one, connective tissue supporting. Third one, connective tissue fluid. Now, the first 2 categories can further be broken down into 2 subcategories, dense and loose for connective tissue proper. And that's like dense regular connective tissue. That's like your tendons and your ligaments, like your ACL. All those tissues connect 2 things, and that would be an example of dense regular. Now, something like dense irregular is going to be more in your skin. It also connects 2 things, but it has an irregular pattern to it. Now, loose connective tissue is like areolar, adipose, reticular. Areolar can be found in the intestines and holding things together in the intestinal region is just one of the locations. Adipose, we're looking at fat cells. So in the subcutaneous layer, the hypodermis, we've got this yellow stuff, that's fat. There's also fat all throughout the body that can be stored as adipose tissue. And then finally, reticular tissue is going to surround something like the spleen or the kidneys in the body. All right, we're on to connective tissue supporting, and this one's going to be broken down into bone and cartilage. So bone and cartilage are the subcategories. And underneath bone, you've got compact bone and spongy bone. Now, compact is more superficial. That would be like this part out here. And as you get in, it becomes the spongy. And compact bone is like just this outer part of it, very, very compact and durable, whereas spongy bone is more in here before you get to the cavity in the middle of a bone. As you can see the difference in compact, I'll put a C on it, and then spongy, I'll put an S, and then in the middle, it would be a space. That's bone. Cartilage is similar to bone in the sense that it really provides support for bones and other things as they come together. First up is elastic cartilage. That's like in your ears. See this ear picture down here? Your ear has elastic cartilage. Take a second, go ahead and feel your ear. You can bend your ear, you can turn it side to side, but you'll feel there's some tougher cartilage within your ear. Specifically, that's elastic cartilage. Elastic cartilage is elastic, flexible, Fibrocartilage, though, it's not as flexible, it's durable, it's tough, it's fibrous. That's like the intervertebral discs in between your vertebrae are made up of a lot of fibrocartilage. And lastly, hyaline cartilage. Hyaline cartilage is in between joints. Now, this is a similar picture to the ACL picture, but the ligament is dense regular. There's other tissue on there that we'll look at, which is hyaline cartilage. And lastly, connective tissue fluid. The only one to talk about is blood. So blood is actually a tissue that connects things. The blood stream, the blood system connects things in the body. All right, let's start with that dense regular and dense irregular tissue. And I've got two on the left that are the same and two on the right that are the same. You tell me which one is dense regular, which one is dense irregular, and why. So go to APR, check these out on Google, whatever you need to do to figure out which side is which. I'll give you about 10 more seconds. All right, hopefully you got an answer here because I've got an answer for you. All right, on the left-hand side, we've got dense regular. Dense regular is going to have what are called parallel collagen fibers. So all these long strands here are collagen fibers. And notice how they all are running in essentially the same direction. Now there's also fibroblasts in there, those black dots, but mainly it's collagen fiber. Collagen, I want you to think durability and strength of a tissue if it has a ton of collagen. Now dense irregular, it also has collagen, but I'm gonna just draw on here and you'll notice there's collagen fibers going in every which direction, all over the place. So it's durable, but it's durable in every direction. Whereas that dense regular would just be durable or most durable in the direction parallel to the collagen fibers. Another knowledge check for you here. Where are these found in the body? Take 10, 20 seconds. Try to figure out where these are found in the body. Don't look at your notes or your book. Let's try to check your brain to see if you're absorbing this information. So take 5 more seconds and then I will reveal the answer. All right, here's the answer. Back one slide and you can see that dense regular is found within these tendons and dense irregular is found within skin or the dermis of the skin. And biomechanically, let's say something like the Achilles tendon, it is very strong in the up and down direction, right? Superior, inferior direction. It's very strong because that's where the fibers are parallel. The collagen fibers are parallel, superior to inferiorly. Same thing with, like, let's say the ACL has its parallel collagen fibers, but if you were to come at the Achilles tendon medial to laterally, side to side, it's not as strong in that direction because that's perpendicular to the collagen fibers. But something like the skin, like I said, has dense irregular, and skin, you can pull on your skin in every single direction and it's going to be strong. So that speaks to the dense irregular connective tissue that has collagen fibers going in every direction. All right, we're on to the loose connective tissue here, and the first main one is areolar tissue. So areolar tissue is going to look like this up here. It's got 4 main structures to it. It's got some things called elastin or elastic fibers, and those are like these thin lines coming across the page. It almost looks like if I were to just draw on it with my pen here, those would be the elastic fibers. But if I were to take more of a highlighter and really just highlight across this slide, those thick bands of pink that you can see going across, those are the collagen fibers. Like I said, collagen is strength, elastic is flexibility. And we've also got the nucleus. Nucleus of what, Professor Klein? Nucleus of the fibroblast. So, oops, fibroblast over here. Here's an animated picture so you can see the fibroblast. But if you saw them over here, it'd just be like a black dot. Notice all these black dots in between everything else. Those would be the fibroblasts. Well, there's only 3 things on these slides, but I told you there's 4 things to know for areolar tissue. So I want you to go look up the 4th thing for areolar tissue and have that ready by lab. Here are the other 2 types of loose connective tissue. First is that adipose tissue, adipose, fat cells. As you can see here, I'll outline all the cells of adipose tissue in the body. Oftentimes they are actually yellow, so not pink and purple and clear, but actually yellow, that fat-like look to it. And reticular tissue, like I said, around the spleen, around the kidneys, that outer, tougher covering, it's going to have some special fibers called reticular fibers. Now, reticular fibers are a special type of collagen fiber. So when you think collagen, also think reticular fibers in a different version or form. So we're talking tough, durable strength as the capsules that surround these organs. But let me give you another study tip, because I got a study tip for the adipose tissue or any tissues, and here is the study tip. Now what you want to do is connect new information to information that's already in your brain. Now how do you know what's already in your brain? Well, as soon as you look at that adipose tissue slide, your brain will immediately think of something. So the question is, if you look at that adipose slide, what does your brain think of automatically? It doesn't matter how silly or goofy or weird it is. What does your brain automatically think of when you look at that adipose tissue slide? For me, it's miniature marshmallows. I look at that and I go, wow, that's like a bunch of miniature marshmallows that you might take and put in your hot chocolate. I love hot chocolate. But the next part of this study tip is take what your brain automatically thinks of and connect it to the new information. So I might say, okay, miniature marshmallows. If you eat a bunch of miniature marshmallows, you might gain some fat, right? You might gain some weight if you ate too many of those sugary things. And that's what this is it's adipose tissue. So connect that automatic information your brain thinks about and the new information. Now that works for me, but you got to pick something that works for you to help you learn this. All right, we're on to the next one here. And if you're keeping track, we've done dense, regular connective tissue. We've done loose connective tissue. We are on to connective tissue supporting. And first, let's talk about bone. All right, with bone, we most often think of a long bone like this. This is the femur. You guys are going to learn skeletal system, all the bones later. But we also have the skull, things like the skull. And today, though, we're going to look more at something like the bone model, or what actually makes up the bone before you learn all the bones later. Now, bone is made up of 2 different types of bone, that compact or more superficial bone, which would be this section here, and the spongy bone inside. So the spongy bone would be like these kind of spider web-like structures. Compact bone is completely tight on the outside. So the question is, which histology slide, top one or the bottom one, is compact, and which one is spongy? Take 20, 30 seconds, jump on over to APR and figure this question out. All right, pause the video if you need more time to figure this out and really give it a try to find this out. But here are the answers. All right, the top one is compact bone. So congratulations if you guessed top one for compact bone. The bottom one is spongy bone down here. These purple things are the trabeculae, so I'm outlining some trabeculae right now with the bone marrow, put a BM for the bone marrow in between those structures. All right, some other ways that I like to remember this is with SpongeBob or sponge here, because we know a sponge actually has holes within it here, and that's exactly what spongy bone has, is the trabeculae would be like the solid part of the sponge, but within the holes of a sponge or spongy bone would be that bone marrow. So bone marrow goes through the holes or the spaces within spongy bone. But compact bone, just like a compact disc, if you guys remember these, you might be too young to remember compact discs for CDs, but there are a ton of them out, and this is like an osteon of compact bone stacked together. All right, let's look at that on the bone model right here. First off, the bone model has a YouTube video, so stop this lecture video and take a look at the bone model video, learn the anatomy, come back. We're going to look about or at a few of the things when you come back. And the first thing you want to do is relate the histology to this bone model. So notice these circles right here. Those are called the osteons. Everything within that circle is an osteon. Just like up here, everything within this circle is like an osteon. An osteon has 5 main parts that we'll look at later. Now, this model can show you an osteon like this, or it can show you an osteon that looks like a tower. That would just be showing you the different levels or layers that make up an osteon. Now, you also see a word called lamellae. Lamellae means layer, but there's interstitial lamellae, there's circumferential lamellae, and then there's also the concentric lamellae as well that make up the osteon. But as you work your way deep through compact bone, one, notice the perforating canals that have blood vessels going through it. Notice the periosteum that covers all compact bone, as well as the Sharpie's fibers that hook it into the compact bone. But if you're deep in bone, you are within spongy bone. And here you can see that bone marrow. Let me give it a BM there, bone marrow. And then this white part, spider web-like, would be the trabeculae. So purple on the slide equals spider web on the model. And whereas this red bone marrow here would be, let me give it a red color for you, that would be this section in here as the bone marrow. So hopefully that helped. And just by looking at this and looking at this picture over here, we've got blood vessels, nerves, transversing through a bone. Oftentimes we think of a bone when someone has passed away and we can see a skeleton. But when bones are in someone who is alive, that bone is alive as well. And it's growing, or it's shrinking, or it's changing shape depending on the stresses that are placed upon it. So just another view of compact and spongy bone here with your osteons as you work your way into the spongy bone with all that trabeculae. Here, you can't see the bone marrow, but you can see the trabeculae down there. Now, the main cell type within bone are called osteocytes. Osteo means bone, cytes means cell. These are the osteocytes right here. All these little dots that have a cell in them would be the osteocytes. Osteocytes. And here's a closer view of those osteocytes, and you can really see the cell shape with the nucleus in the middle. But there's something else called caniculi. Caniculi, and these are these little narrow pathways connecting osteocytes. So caniculi is a narrow pathway connecting osteocytes in the osteon, and you can see them right here. These would be some caniculi that I'm drawing from osteocyte to osteocyte. But osteocytes were not always osteocytes. They first began as osteoblasts, which form bone formation, and then they become osteocytes trapped in the bone matrix, and then what I refer to as retired osteocytes would be osteoclasts that break down bone. And like I said before, don't just use APR. Once you use APR, go to Google, try to find some pictures that look similar but are a little bit different. As you can see here, here's another picture of compact bone, perforating canal going through it, spongy bone over here with the marrow, the trabeculae, again, related to multiple perspectives for these tissues. Study check time for you. Let's look at an osteon, and this is something you'll really want to know, so let's take a minute on it here. If you look at this osteon, and to me it looks like a tree trunk. So each one of these tree trunks is an osteon, but there's 5 parts of an osteon. All 5 are listed down here. Now we've talked about concentric lamellae as the layers that surround or make up the osteon are the concentric lamellae. The osteocytes would be these black dots. So see all these black dots here? Those are the osteocytes. And we said the caniculi connect the osteocytes to one another. My question is, what is the central canal and what is the lacunae? Look that up now. What is the central canal and where is it at on the slide, as well as what is the lacunae? All right, pause the video if you need more time, but I'm going to go through the answers here. The central canal is the center part of the osteon. That's the central canal, also called the Haversian canal. All right, so I wrote it out there for you. That's going to have the blood vessels going straight down the osteon. And if you look at the model right here, let me make my picture larger for you. Here you go. You can see that central canal filled with a blood vessel. It's going vertically into the osteon. And the last one, maybe the toughest, is the lacunae. You can't really see the lacunae on here because the lacunae is like a cavity or a divot to where the osteocyte would lay. All right, so what do I mean by that? What I'm looking at here is the black divot would be the lacunae. So this dip into the ground or into the osteon would be the lacunae. The red part would be your osteocyte. So the osteocyte sits within that lacunae as it shapes around it like that. Now technically this is a cut, so the lacunae would go full 360 there, so you can think of it as like the egg shell would be the lacunae, and then the egg yolky part in the middle could be the osteocyte. And lastly, when you're talking about bone, you've got to talk about osteoporosis and the degeneration of bone, compact bone, spongy bone. You can see it becomes more and more porous as you go from left to right. And this bone over here is not strong at all. How do you fix this? You need calcium, you need vitamin D, and you need stress on the bone. Stress like a running workout or something like that, but then you've got to rest afterwards. That will stress the bone. Then you replenish it with calcium and vitamin D, and then it grows stronger, and then you stress it again. So running, jumping, hiking, weight-bearing exercise can keep bone strong. All right, enough of bone. We're on to cartilage here. Let me show you all the cartilages. We've got that elastic in the ear, fibrocartilage in the intervertebral disc, and hyaline cartilage in the joints. But first thing you got to know is anytime you see the word cartilage, I want you to think of chondro. So chondro equals cartilage. That's what chondro stands for, whether it's a chondroblast, chondrocyte, those are cartilage cells. And first up is elastic cartilage. So elastic cartilage, as seen here, is going to have all these chondrocytes to them. So you see all these circles. Let's circle a few for you. These would be the chondrocytes with the nucleus in the middle. But notice, if you look real close, and this is just the elastic cartilage in between the black lines, you'll notice some elastic fibers running through it. You gotta zoom in almost as far as you can to see these, but you can definitely see them over here as well. That's the elastic fibers that we said make it elastic, make it flexible for things like the ear, or something like the epiglottis. As you can see here, that's in the back of the throat. Next up, fibrocartilage. Fibro means fibrous, tough, durable. You're still going to have some chondrocytes, but instead of elastic fibers, you're going to have a ton of collagen fibers. Collagen equals strength, so you got all these collagen fibers, these blue wavy lines right here. This is collagen, collagen. Let me actually use what I used earlier with collagen, and I'll draw in this thicker structure, right? Because collagen is thicker as it travels through fibrocartilage. So definitely know intervertebral disc, something called the pubic symphysis is where fibrocartilage is in the body. And the last form of cartilage is hyaline cartilage. Hyaline cartilage is going to be found in between the joints. It's a thin layer of cartilage, sometimes called articular cartilage, when it's in between joints. As you can see here, this thin layer in between the bones would be articular cartilage. This is protecting the bones from rubbing together. If you rub two bones together, it's going to produce friction and damage. That is not good. And maybe you've heard of former football players or athletes or whoever it might be that says, I have knee pain or back pain because I don't have any cartilage left in my joints. If that cartilage breaks down, then yeah, you're going to get more bone-to-bone contact. And before I move on, I want to dispel a myth that is out there. As a long-distance runner and a researcher, I hear all the time that my knees are going to break down and that running is bad for your knees, and that simply is not true. The research, especially this research study here, talks about long-distance running versus those who are sedentary. And if you scroll down, we're talking 50 to 17 or 72-year-olds. And what they did was they found less or the same amount of osteoarthritis in runners versus sedentary individuals. And actually, they found a lot of other issues in those with sedentary lifestyles. So as the conclusion says here, long distance running in healthy older individuals was not associated with accelerated radiographic osteoarthritis. So this is great data here as it supports running. So keep running, walking, hiking, jumping. As long as you do it in a safe way with plenty of rest, it will actually strengthen your joints and your hyaline cartilage. All right, getting to the end here, and we're actually on to muscular tissue. Last part of connective tissue here is the blood. So for the blood, this would be like within a blood vessel. You guys ever been inside a blood vessel before? All right, here's the blood vessel model. Let me take you inside of here. Whoa, here we are inside a blood vessel. These are the red blood cells floating around, erythrocytes. And this is our final type of connective tissue, is blood. Later, you're going to learn all about blood, but for now, we want you to know what a peripheral blood smear is. This is, let's say you do that finger prick, and if you haven't figured out how to do a finger prick or watch that video yet, check out the finger prick video. But let's say you take a drop of that blood and you put it on a microscope slide, take it on over to the microscope here and put it underneath, and what you would see is this peripheral blood smear. So all these pink dots are red blood cells. These are actually pink and red in the body when they're filled with oxygen. The white blood cells are leukocytes. Those are a little bit larger and not as common. There's so many red blood cells, millions and millions, over about 20 or 30 trillion in your body total, versus the white blood cells, which is only about 30 billion. So much difference between 30 trillion and 30 billion white blood cells. So blood smear, the word smear literally means to smear blood onto a slide and then look at it underneath that microscope. All right, if you're keeping track, we're done with connective tissue here. We did epithelial, we did connective. Now we're on the muscular tissue right now. All right, 3 types of muscular tissue. We've got skeletal, we've got cardiac, and we've got smooth. Skeletal, we're talking about muscle that moves your body, biceps, triceps, calf muscles, all those things you'll learn later, but that is the skeletal muscle there. It's voluntary and striated. What's striated mean? Striated means these lines right here are striations. They're produced by an overlap of what's called actin and myosin. So this is a sarcomere right here, and the sarcomere is what makes up skeletal muscle. So whenever you see an overlap of the red and the blue, that would appear as a striation. However, in between you've got just the blue, and thus there's kind of a lighter color to that area. Now something like cardiac muscle though, that's going to deal with the heart. This one's also striated, as you can see the striations, whether you see an animated picture or the actual cardiac tissue, but it also has something special within it. Here's what's special. What's special are these things called intercalated discs. Intercalated disc, what are intercalated discs? These are intercalated discs right here. If you zoom on in, you can see there's these little lines that make up connections between the cardiac cells, cardiac tissue. All right, much better. Now we can see things like the intercalated discs. They're different than the striations. So you still have striations here, but the actual intercalated discs would look more like this on some histology. So a big long line connecting the different cardiac tissue. And stick with me for one more because we've got the smooth muscle. This one's all throughout the body, but be careful. You've got to look just within this area. This is where the smooth muscle would be. This is more epithelial up here, connective tissue down there. So make sure you're looking just at the smooth muscle. Smooth muscle, it's not striated. No striations going on with smooth muscle. And this really is responsible for pushing food through the GI tract, the gastroesophageal tract, digestive system, whatever you want to call it. That's what smooth muscle does rhythmically. That's called paracelsus. But there's more places where you have smooth muscle within an artery and a vein. See this muscle right here? That's smooth muscle. Well, let's go to skin. There's also a smooth muscle called the arrector pili muscle in your skin. This is connected to the hair follicle and the hair, and when you contract this muscle, which it does it involuntarily, because smooth muscle is involuntary, it stands your hair up. That's when you have goosebumps. So no examples for skeletal, no a couple examples for cardiac, and definitely a few for smooth muscle as well as this information here. You want to know that skeletal muscle is voluntary as well as striated, voluntary as well as striated. Cardiac is involuntary. Don't have to contract it with your brain consciously. It just contracts on its own about 72 times per minute. But smooth muscle is also involuntary, but it's not striated. Again, striations is the overlap of actin and myosin within that sarcomere. All right, last type of tissue that we'll look at here is nervous tissue. One slide for you. Nervous tissue, we're talking about neurons, the brain, the spinal cord, and all the nerves that travel out. If you didn't have a nervous system, nothing in your body would work. So this is a very important type of tissue. But for now, just know this slide and a few general things about it from APR. All right, take a second. That was a ton of information. Come to a blank piece of paper, write all this stuff out, take some time to study, and learn all the different types of tissues in the body. I want to finish this video talking about the Semmes Weinstein Monofilament test. This test is going to use these instruments here, the monofilaments, and really it's a test for diabetic neuropathy. Anyone know what diabetic neuropathy is? We've all probably heard of diabetes, but have you heard of some of the complications or damage that diabetes does to the body? It's an extremely damaging condition to the body, and one of the things that can happen is neuropathy here, nervous system, pathy, disease. So basically, it's the diabetes attacking and destroying the nervous system. And when this happens, the nerves and the sensory areas of your skin no longer function. So this is a test where we're going to push this thread. Let me get one you can see here. Push this thread against someone's foot, and if they can't feel it, then that is a positive Semmes Weinstein test. Or rather, we'd say it's normal versus abnormal. Abnormal responses, you can't feel it. If you can't feel it, the nerves have been damaged. Watch this full video down here. This will walk you through the Semmes Weinstein test. You're going to try it at home using some other materials other than the lab materials, but watch the video and make sure you know what type of receptor is being stimulated when you do this test. Well, bonus type of tissue here for you. What is this?
Scar tissue that I wish you saw. Sarcastic, Mr. Know-It-All. Close your eyes and...
All right, we haven't talked about scar tissue. What is scar tissue? Well, scar tissue is when you damage the skin and then you have replacement collagen fibers that swoop on in and pull the skin together and form a scar. Scars are permanent. It's not another type of tissue, but they do call it scar tissue. It's those collagen fibers that are really tough that hold it together.